Hello everybody, my name is Professor Frank Gunn-Moore and I've been asked to give you a short talk about the research that we're doing, which I've entitled The Neurobiology of Alzheimer's Disease, and with a subtext of why do only humans get it? So I've been working on this topic for over 20 years now, and what I always try and do is I try and give you sort of like simple questions, which most of you will know, they're always the hardest to actually answer. I'm going to give you some new ways of looking at things, the approaches that we take. Hopefully I'm going to show you some things that you may have never seen before. And as ever with the sort of research, you need a large slice of luck. So the global impact of dementia is enormous. I mean, obviously we're dealing with another sort of very serious issue at the moment. But we mustn't forget that there are other issues that we've got to try and deal with. In the case of dementia, this is a rapidly sort of growing problem for all of us. At the moment, it's predicted that by 2050, we'll have 131 million cases. It's also a problem for the whole world. It's not originally, people used to think of it, tend to be as like a European and North American issue, but we now know that it's a problem for the whole world. Going further, we also realize that either you may be unfortunate enough to actually sort of succumb to dementia, or you may have a family member who actually will develop dementia. However, if you're lucky in that neither you nor your family actually develop it, you're still going to be affected because it's become a huge sort of financial issue for the world. So in 2018, it's now costing $1 trillion per year to actually look after people with dementia. And that essentially, when I'm talking to politicians, puts it at the sort of scale of being the 18th largest economy. It's actually at current present bigger than Apple, bigger than Google. So this is a problem that's not going to go away. And so we have to actually tackle this. So how are we going to do this? Well, the first thing you need to know is actually literally how sort of the brain works. And roughly speaking, we can actually characterize the brain in that it's got different regions of the brain that have different functions. So if we start at the bottom here, so the cerebellum is the part of the brain that controls your coordination, balances and reflexes. The occipital lobe is involved with vision. The parietal lobe is involved with touch perception and the integration of senses. Temporal lobe deals with hearing and memory, and then your brainstem deals with breathing, heart rate and balance. The area of the brain that I haven't mentioned so far is actually the frontal lobe. Now the frontal lobe is the region of the brain which is involved with your consciousness, essentially makes you what you think you are as a human being, your emotion and your memory. And unfortunately this is the region of the brain which is affected in Alzheimer's disease and pretty much all forms of dementia. However, as ever the optimist, I always think that this is one of the areas of the brain that will actually help us overcome this problem, because this is your region of the brain that gives you your creativity, gives you your imagination. And as such, this, though it may be affected, I do feel that this area will actually be our greatest savior. Now, to put it in context, human beings are unusual because human beings have a very elaborate sort of frontal lobe. Other animals have these regions of the brain, and to some extent other animals do have a frontal lobe, but ours seems to be much more sort of advanced than other animals. I'm always asked about, well, what's the difference between dementia and normal aging? And to put this in context, this table is quite useful in that a lot of people get very worried when they sort of think, well, maybe I'm getting dementia. There is no doubt that as we get older, things do change. So in normal aging, these are sort of common sort of factors. Not being able to remember details of a conversation or event that took place a year ago. Not being able to remember the name of an acquaintance. Forgetting things and events occasionally having difficulty finding words. And you're worried about your memory, but your relatives are not. Now, in the case of dementia, it's different. So here, it's not being able to recall the details of recent events or conversations, not recognizing family members, forgetting things very frequently, and frequent pauses and substitutions when finding words. People with dementia are sometimes very good at trying to actually sort of fill in the gaps. And rather than you're worried about your memory, it's your relatives that are more worried. Now, in the case of dementia, there are probably, we now think, about 75 different types. But the most common form that you will have heard of is Alzheimer's disease. And this is about 60 to 65% of cases. Now, the post-mortem hallmarks of Alzheimer's disease have been known for many years. So Alois Alzheimer, when he first saw this is actually over 100 years ago, saw that there were these chemical changes, what he referred to as amyloid-containing plaques, and these were actually occurring outside of the cells, and then the formation of neurofibrillary tangles which were occurring inside of the cells. So we had a biochemical event occurring outside of the cells, and then also an event occurring inside the cells. The issue of this, of course, is that, well, this is, of course, what we can see when somebody's died. These are post-mortem tissue of somebody's died of Alzheimer's disease. And as a result of this, the real question is, well, what actually happened 10, 20, if not 30 or 40 years before this? And the biggest problem with this field has always been the fact that your brain is encased in bone. It's very difficult to actually see through bone to the level of resolution that we really need to see. So to be able to tackle something like Alzheimer's disease, I really truly do believe we need to have disciplinary research. And that means taking both biology, chemistry and physics to actually look at this problem in a unique way. I also like to think as well, a lot of the research we've done, we initially started off looking at the mechanisms of what's actually occurring in Alzheimer's disease, but that's now led us to interesting things to find out what's occurring in diagnosis and even sort of like treatments as well. So my starting point. Well, I started this work actually when I was a postdoc in about 1997, and I became interested because this substance amyloid that I was telling you about, everybody had always sort of told me, well, it occurs outside of the cells. And so most people have done sort of studies saying, well, amyloid, we know it kills neurons and it kills cells, but how does it do this? Well, one way would be to attach to a cell surface target receptor, something on the surface of the cell that amyloid attaches to and then elicits some sort of response. But I started to realize, well, there could also be a way that amyloid inside of the cell could also be toxic. And that's because actually amyloid is first made inside cells. So what are these targets that are in the sides of the cells that can cause the death of these nerve cells? So why would this be important? Well, this is a sort of bit of a thought experiment. Well, because toxic amyloid is first made inside the cell. These amyloid plaques that we've seen outside could actually be quite late stages. So I used to think, well, is that the early stages of Alzheimer's disease? So how could this internal amyloid kill cells? Well, over the years, we found a number of targets that it attaches to. One of them we called, not very imaginatively, amyloid binding alcohol dehydrogenase, or ABAD. ABAD's normal sort of function in the brain is to give you energy. And that's important because ABAD is found within mitochondria. So most of you probably have heard of mitochondria now. These are the structures within all of your cells that use food products to make energy. So a lot of the time you can use glucose, but other times you have to use other energy substrates. And how do you convert that into energy? Well, that's via the mitochondria. So we knew that this ABAD is inside mitochondria, and here is actually ABAD tagged with a green fluorescent protein, and you can see these little green dots. These are the mitochondria. Here in the center we have an electron micrograph of what a mitochondria likes, and so the question was, amyloid attaches to ABAD and can lead to cell death. We'd sort of shown that, but of course the question is, how? Well, here are my embarrassing slides. As we age, things change. So when I was much younger, in this rather stylized sort of jumper, as you can see, my brain was very good at just using glucose as its energy source. But as I got older, and you can see different sort of like substrates I was being introduced to, this changes. So your brain initially, as I say, starts using glucose, but as we get older, your brain uses less and less glucose as its energy substrate. And the way it does that is because it uses mitochondria, and it uses things like ABAD to produce the energy. So as we get older, there's a decrease in the amount of glucose that your brain uses, but an increase in mitochondrial function, or the need for mitochondrial function. And we know this is true because if you do a PET scan, so PET scans are looking at glucose metabolism in the human brain. You can see as we get older it changes. And people with dementia, and in particular Alzheimer's disease, the amount of glucose they use goes down even more. So our studies had sort of identified that amyloid attaches to this target called ABAD. So we found out exactly what the shape of our ABAD was. Now, I know for many of you this won't mean much, but this is actually what ABAD really looks like. And there's a part of it, referred to as loop D, where we knew the amyloid was attaching to it. The result of that is that we knew that this was actually sort of causing cell death. So, as I said, the question was, how was it actually doing that? Well, we found out that when amyloid attaches to ABAD, it actually produces various toxins. These toxins are made inside the cells, which then stimulate particular signals, which then influence the genes which are being switched on and off in sort of nerve cells. So then the question is, well, what are these genes doing? What are these genes that are being switched on and off? Well, what we found was that a lot of these genes were actually changing how nerve cells actually talk to each other at what we refer to as synapses. So here we have two neurons or nerve cells, and here is the synapse, and information would be passing from one nerve cell here to the next. What we found was that these genes that were being switched on and off in Alzheimer's patients, and we showed they really were occurring actually in human brains, were actually changing how these synapses work. And this is where actually I sort of did some public outreach work with Sally Magnuson. Some of you may know her. She actually published a book about her mother who actually died of Alzheimer's disease, and she came to see me to try and understand some of the basic biology that was going on. And at one moment in the lab I was showing her sort of pictures of nerve cells talking to each other, and I sort of said to her, well, you see these connections, these synapses, this is where memories are actually held. And so then she thought, well, in course in Alzheimer's disease what happens? Well, those memories are lost. And as such, it's because you're losing these synapses. And that's actually why she called her book Where Memories Go. So we've shown sort of already at the moment changes in the energy production of nerve cells in mitochondria are causing how synapses, how memories are actually changing. So that could lead on to the question, well, could we cure dementia or can we cure Alzheimer's disease? Now the problem is that it becomes a question of which target. So neurodegeneration is another term for what we're going to call dementia here. And the issue here is that there are lots of different ways that we can lead to this neurodegeneration. I've just told you about mitochondria. Some of the other proteins that I've been involved in are actually in protein handling disorders or maybe growth factors. And so it becomes a combination of which targets. I would say to you, one of the things that's important to remember is that it will probably never ever be just one drug. It will be a combination of drugs of what we need to put together to actually cure neurodegeneration and dementia. Well, in the case of our ABAD, if you remember, I told you we identified where amyloid actually attaches to ABAD. And so what we did was that using a mouse model for Alzheimer's disease, we'd found, and as I've already said, various genes were being switched on and off. We then gave them a region of ABAD, that region called loop D, where the amyloid attaches, as like a decoy, so bind to me and not to ABAD. And when we did that in these living animals, the genes actually that we'd identified that got switched on and off actually got switched back off or on, depending on which way they were before. More exciting than that was the fact that these animal models, they actually have problems with their memory, which of course is one of the issues in Alzheimer's disease. When we gave them this decoy, their memory improves. So of course what that means is that we've essentially found a potential drug target. And we're trying various methods to make an improved therapeutic compound. Now this takes a long time. At the time when we first published this, as you can see, we got lots of press, but as I always told them, this will take time. But we are having a go. And that's because where we start to move into the chemistry of things. So to cut the long story short, we have now screened over 350,000 compounds which actually have activity to our ABAD. We've had to actually invent completely new ways to actually test them as well. And I'm happy to say we're about to test them now. We're starting to test them in biological systems. Obviously, I'm not allowed to show you what some of these structures are, hence you've got the smiley faces. One thing to realize, that took me 20 years of work to get to this point. Anyway, I now want to actually sort of push your imagination and tell you about some of the physics and how that's relating to our work now as well. And this is all came about because light and how light can be light has been studied in St Andrews for many years. So Brewster invented the kaleidoscope. Alan here, he was a lovely chap. He did a lot of the early pioneering work on LEDs. And Wilson Sibbett invented what we call the mode locking lasers, which was a fantastic achievement. So the following of what I'm going to tell you all came from one email. So one of my sort of colleagues, and it was actually Wilson Sibbett and his friend and my friend Kishan Dalakia, who basically sort of said to me one day, we can do strange things with light and lasers. Would you be interested in working on them? And I sort of said, well, why not? And so just to show you, light can do some strange things. So one of the first things you can find out is light has momentum. So these are very small spheres which are all being moved by just using light. So these are actually 1 micron spheres. They're tiny. And as a result, I always like to joke at this point, this is the world's smallest golf swing. So nobody touched them. That was all being moved, as I say, by light. So that's a cute sort of like movie. But can we use that on a biological system? Well, the movie done here is what you're looking at here is the growth cone of a nerve cell. So this nerve cell is growing, but that little spot there is us guiding it using a laser light. We can actually get this nerve cell to grow towards a laser light. How does this work? Can we expand its use? Can we control this? To this day, we do not actually understand what the mechanism is behind this. But as you can see, you can start to really sort of explore completely new parameters. One of the nice things about this sort of work is it's showing us that cells don't just respond to chemical cues, they also respond to physical cues as well. Going on further, one of the things we actually sort of developed was that we can actually control and shape light. And in this case, what we could do was we could make specific little holes in cells, what we refer to as the optical syringe. That allows us to put drugs and compounds into cells. So here, what you're actually looking at are nerve cells. These are nerve cells which have actually been developing from the front part of the brain. And then what we can do is it's a touchscreen system. You can touch the screen and then you're actually making a little hole. We're guiding a laser to make a little hole in that cell to then put whatever gene of interest we'd like. And in this case, what we put in was a fluorescent protein that made nerve cells go red and a separate one where a nerve cell went green. All of this technology we made so simple. So literally what you can see here, we were demonstrating to a particular politician how we could actually sort of just anybody could use this technology. We also took this technology so that we could also put it on the end of an optic fiber, which is literally the size of a human hair. You can start to see where you might be able to actually utilize this in sort of unusual ways. Going on further, the use of light has been revolutionized by identifying a particular gene product which originally came from a small type of algae called Chlamydomonas. Now these little algae, what they can do, you will find this in your pond water, they will actually, if you shine light, they will actually swim towards the light. Well, why do they do that? Well, that's because they have these gene products, these proteins that sit in the membrane of the cells, which when they respond to particular wavelengths of light, they open up. And they open up such that then either sodium ions or maybe chloride ions come through. Now, what is possible to do then is possible to take the genes for this and now encode that into a mammalian nerve cell. So now when you shine blue light, these cells become electrically active. And that's quite remarkable. Previous to this, the only way you could stimulate nerve cells was by essentially using electricity. What we're talking about here is that you can stimulate nerve cells by just using light. So you can then ask the question, well, could I build a memory from light? And extraordinary enough, it is actually possible to do that. It's actually, we didn't do this ourselves, but it's been done by groups in America. They've shown that you can create a false memory by using light to stimulate specific nerve cells. Going on further, this is our own work. What we've actually done here now is used another one of these optically sensitive proteins, and we've put it now in a complete network of neurons. And now what we can do is that we can just stimulate, as you saw the blue light that was flashing up here, just these nerve cells here. What we're now looking at is actually how much calcium is being stimulated, being produced in these nerve cells. That leads to the capability of, rather than just studying one single nerve cell at a time, but we can work out a complete connectivity map, how our complete network of neurons are actually being stimulated. And you can see we actually only published this a couple of years ago, and we've had huge interest in this because, of course, this might be possible to use in drug discovery aspects as well. Going on further, we can now see the electrical activity in nerve cells using light. So before we were using light to actually sort of like stimulate nerve cells. Here what we're seeing in this movie, and this again I should say it's not our work, but we can do this in a lab, is they stimulated in this nerve cell, and now you're seeing the electrical activity of that nerve cell. I'll run that one more time. Any moment now, stimulate, and that is an action potential. That is essentially forming memories. Quite extraordinary. We've gone on further with our ability to shape light. We've developed new types of microscopy. This is the capability of most microscopes shape the beam in this sort of form, but we've essentially able to make big, long sort of like strands of light and even make curved light beams. The reason why I actually am showing you a picture of this chap is this is Eric Betzik, and he actually sort of also agrees with us that this type of what we refer to as light sheet microscopy is the future. He won the Nobel Prize. The result of our studies is that we published a very high impact paper so that we've now been developing these new types of microscopes. And in particular, by tying up with the company M Squared, we can now image large pieces of brain in one image to a fantastic depth. This is, quite remarkable for myself. As you'll see these two movies running through, you can see incredible depth, but in a large volume. We hope it might be possible to image a whole mouse brain in just 7 images, 7 clicks. One of the things I'm showing you all about this is that none of this technology existed 10 years ago. I'm asked all the time, are we going to cure dementia? Are we going to be able to solve the problem? To some extent, I am an optimist because you never quite know what's going to be around the corner. Now, as some of you may have been sort of remembering at the beginning of this lecture, I asked a question, why do only humans get dementia? And I've told you about all our technology and the chemistry and biology and physics that we're doing, but I haven't really answered that question. Well, I'm going to take you back. So if you remember, I talked about the post-mortem hallmarks of Alzheimer's disease. And in actual fact, there are 3 different changes that you see. One is these amyloid-containing plaques that I told you about. One of them is a change which I haven't had time to talk to you about neurofibrillary tangles that occurs within the cells. And another one is lipid changes or changes in the fat. Over the years, a lot of our work has actually identified lots of genes and proteins that are changing in Alzheimer's patients. But what about other mammals? Well, all of this work I'm about to tell you started with a conversation with my lovely wife, who happens to be a professor of feline medicine at the University of Edinburgh, and we were discussing one of our cats called Cardew. I should say our cats have always been named after very good malt whisky. Now, Cardew was quite an elderly cat at the time, and we were discussing one morning. I sort of said to Danielle, well, he seems to be sort of behaving a bit strange. He seems to be forgetting things. As a result, I said to Danielle, do cats get dementia? And she said, well, in veterinary terms, we don't call it dementia, we call it cognitive dysfunction syndrome. And these are, if you're a cat owner, you will actually sort of probably agree with a lot of these changes here that you can actually see. So we then started to embark on some studies actually together. And what we had was Danielle had a series of animals that had come to see her in the clinic who had died with neurological problems, and they never knew why they had actually died. Well, by looking at the brains of these old cats, we were able to stain for these changes that we normally see in humans. So in Alzheimer's disease, amyloid plaques is what we see, but in cats, you don't tend to see it, not lots of spots here. Dogs, however, and that had been reported by other people, did seem to show this amyloid change. However, if you looked at this tau story, so this is the these neurofibrillary tangles that are occurring inside the cells. Here is the humans. You can see this the staining here. Here's the dogs. Doesn't seem to do sort of like the staining inside, but cats do. So what we have here is our old cats are showing one of the signs of Alzheimer's disease, and dogs show one of the signs of Alzheimer's disease, but they don't, in both cats and dogs, they don't show both that you see in humans. That work we sort of published, and people got very interested in it at the time. Subsequently, a collaborator of ours who actually sort of wrote this very good sort of paper on the comparative pathology has actually now sort of collated the changes that you see in the amyloid here, the AP, all these intracellular things, these neurofibrillary tangles. And what you'll see if you look at all these different species is that some of them, yes, do the amyloid. And here's in our case, our cats here, they do sort of not really the amyloid, but they do substantial sort of like changes in the neurofibrillary tangles. Or they might do the other way round. There are some animals that had been reported, obviously, to sort of like change both. In the case Wolverine, my wife got very excited about that, but I think she was thinking about Hugh Jackman at the time. But in the case of Wolverine, I don't think that's ever going to be a particularly good animal model. One thing to note is that here, mice and rats, which people like myself, of course, have used for many years, sort of like as models for sort of like dementia, you can see they don't seem to do either. What we have to do is genetically change them to show the hallmarks we see in humans. So that in itself was quite an interesting sort of publication, but I came back into this field more recently because of a particular talk I heard. Now, this talk was given by Sir Professor Simon Lovestone at Oxford. He was giving a talk, I think it was actually in Dundee, which was entitled Dementia, Diabetes and the Killer Whale. Now, as I've got older, these are the sorts of talks I like to go to. In his talk, he actually sort of said, well, he'd come up with a new sort of hypothesis about sort of why humans got Alzheimer's disease. And he said, well, the unusual thing we have is that we tend to live quite a long time, but we stop reproduction quite soon. And as a result of that, he sort of said, well, other animals that do that, maybe they also get dementia and Alzheimer's disease as well. And the one that he talked about was killer whales. So killer whales can live 70, 80 years and actually stop reproduction, usually in their 40s or 50s. That triggered a memory for me, because years before, after I'd published the studies on cats, this lady here, Maria, was actually doing her PhD at the time, and she sort of said to me, seen my sort of publication, and sent me this bit of data she had, that she'd seen where she could see amyloid and she could see neurofibrillary tangles occurring, so both the extracellular and the intracellular events in something called stenella. Now, stenella, if you don't know already, are dolphins. And these were actually dolphin strandings that she'd actually sort of been working on and staining in the same way that we do with humans to see what was the pathology, and found both amyloid and neurofibrillary tangles. And obviously when we published this, we got an awful lot of press on this. So I will make you a prediction. Humans, guess what, are not the only ones who get dementia. There are other animals as well that can do this. A lot of the reasons why we've missed it to some extent is just we just haven't looked hard enough. I can tell you, and I haven't published this yet, we are now finding this in other species of dolphins as well. So that leads me to pretty much the end of my talk. One of the things I want to leave you with is that all of these studies have taken me a long time. As I said at the beginning, I started all of these studies in 1997. And academia is not a sprint, it is a marathon. I've hopefully tried to show you that research can come from you lots of different angles. I'd like to sort of think that when I first sort of had this technology trigger, this is called the Gartner cycle. You then get what we refer to as the peak of inflated expectations. Everybody gets very excited. You then can go into the trough of disillusionment, where everybody tells you you're wrong. And then hopefully you get into the slope of enlightenment. And now I'm hoping we'll get into this plateau of productivity. My last slide is to conclude this talk. To conclude, to tackle this disease, We use biology to understand the biochemical changes. We use chemistry to make new drugs. And we're using physics to develop new technology. As I tell all of my gays, and there they are all waving, never be afraid of the approach. That is the most important thing in science, and imagination is critical. I hope you've enjoyed my short talk, and I shall stop there. Thank you.